Hi, I'm Seth Wannemuehler. I'm a new faculty member at the University of Minnesota, helping to take over some of the Rose research, and excited to share some of the upcoming publications and things that the team has been working hard over the past several years for. So with that, I'll let David take it away.
Thank you, Seth. Can people hear me?
Yes.
Oh, perfect, perfect. And I see that you can see the screen. So we're excited to share updates on the phenotyping work that we've done with different races of black spot and roses to form the foundation, the validation set to be able to more efficiently develop and validate markers for these different qualitative black spot resistance genes. So the rose group here in the upper Midwest has been working with roses for a long time. And so over the years, some folks have come and moved on to other great positions in life. And so Stan recently retired, and Seth, as he just described, is a new faculty member in the woody plant breeding position that Stan used to hold. And I started working with Stan years ago, about 25 years ago, with my passion for rose breeding and roses on work focused on black spot resistance. And Deanna, who's in the upper center there, she was a postdoc for a few years here, paid for through the the grant here, Sustainable Roses. Emily Moore is a recent master's student who just graduated. And then, of course, Seth. So the work that we're presenting has been funded by multiple sources here depicted on the lower part of the screen. So for just a little bit of a review here, I've been coming in and out of classes and listening as I can, so I'm not sure exactly how much black spot has been talked about except for some field screens. It's one of the most recognized plant diseases around the world. It's caused by a fungus by the name of Diplocarpon rosae. It's a hemibiotrophic organism, so it can live on both dead and live material. So it could overwinter on dead leaves and debris and things. We could grow it on artificial medium in the lab, even though it doesn't grow very well. It can make multiple cycles per year. So from infection to sporulation in the lab under perfect conditions, we could do that within a week. But typically outside, it takes a little longer, but that's many cycles throughout the growing season if the environment is conducive with lots of rain and moisture and the right temperatures. So it leads to defoliation, weakened plants, less photosynthase, less flowering, less beauty. So over the years with our group in the upper Midwest here, we've identified 4 different qualitative resistance genes. And on this table here, we have 4 rows depicting the rows in the left-hand column that we found that gene in, and the next column in purple, the RDR gene. So RDR stands for resistance to Diplocarpon rosae, and then just in sequence as they've been found. So in Germany, RDR 1 and 2 were found, and then we've identified 3, 4, 5, and 6. As at least unique places in the genome, some loci or locations where there seems to be a major gene or qualitative gene located, even though we haven't gotten to the gene level per se. Across the top here are races 1 through 13. We've identified and have well preserved 13 different races of the fungus, and each race can uniquely either infect or not infect various roses. So what we have depicted here in the table are the races that the particular rose and that gene confer resistance to. Like for instance, George Vancouver with RDR3 is resistant to race 1, race 2, race 4, 5, 6, and so on, but is susceptible to races 3, 9, and 10, and 11. So these resistance genes are dominant, which is great. So if we can pyramid them or combine them in new roses, we can get new hybrids that have resistance to a larger and larger ratio or percentage of these races. And some of the combinations of RDR genes can even confer resistance to all 13 documented races. So where are we at with phenotyping? So that's what I've especially been focused on at River Falls, which is just about a half hour, 45 minutes from University of Minnesota. And last summer, the American Rose Society gave us a grant to pay for some student labor and some laboratory resources. So Leah on the left, Elena in the middle, and Leah's twin sister Michelle helped with the project, which was great. So we've done a lot of inoculations in the lab. A couple years ago, I shared about what we do for detached leaf assays, and I'll briefly just describe them here today. So we take leaves off of roses in the greenhouse where there's no black spot. We need at least 6 to 8 hours of direct moisture for the spores to infect. So we get these beautiful leaves from the greenhouse. We wash them a bit in the lab, put them in those clear boxes with moist paper towel to keep them from drying out. And then we prepare inoculum of each of the races. We keep things separate. And and here's just an image of what it looks like under the microscope with a hemacytometer. And we count how many spores are in a certain grid, and then we dilute the solution to get between 30 and 80,000 spores per milliliter. And then we put those droplets with spores on our leaves. And then within 2 weeks, we could see if it's a susceptible interaction between that rose and that particular race or a resistant interaction. And we gather that data over time to look for patterns for what races particular roses are susceptible or resistant to. And there's unique patterns, as we've seen, for each of the RDR genes that we've characterized so far. And then these roses become very instrumental to validate future markers to get really close to the gene of interest to recognize how useful our markers are. So last year at the update meeting, I shared where we were at. We continued to work through the end of the semester, looking at more and more roses. So we have several hundred roses that are phenotyped to this point. Most all roses are susceptible to every race, which is interesting. And then those that do have some resistance to specific races are especially valuable for us to use to validate markers for the different RDR genes. We've worked with 6 different races. We have the specific isolate listed here. ACT is isolate of race 8, BEP, isolate of race 12, and so on. These 6 of the 13 races are particularly informative. They grow well for us and are the ones that we focused on. The red boxes are a susceptible reaction. The green boxes are resistant. And what I have depicted here are some unreleased roses from Will Rattler's breeding program and some from mine, and then lots of cultivars as well. This summer, I didn't have any monetary resources to work with, so I volunteered and had some greenhouse space at River Falls, and I brought in a couple hundred roses to be able to work with and continue phenotyping some newer cultivars, relatives of those that we know have particular RDR genes. And then this fall, the American Rose Society renewed the grant, and now I'll have some resources to continue validating markers and phenotyping. And what I'd like to do now is transition to where we're at with RDR3, and then Seth, I'll continue on with RDRs 4 through 6. So RDR3, we've been studying for many years, well before this grant. It confers resistance to 9 of the 13 races, which is great. And one thing I'd love to mention is that race 8 is the most widespread race across North America from some past work that Vance Whitaker did for his PhD thesis. And we've been able to find these additional RDR genes because we see roses that aren't getting infected in gardens like many of you are hosting these trial sites, whereas neighboring roses do get infected. So we know there's some sort of resistance there that's unique. And race 8 is the most widespread race. And all the different RDRs that we found so far tend to confer resistance to race 8, and that's part of just being able to identify that there's a unique resistance to chase. So what we did previously with the Ross Breed grant is what Jason Zern do some mapping with the Axiom SNP array with a population derived from Morden Blush, which is susceptible to all the races, and George Vancouver that has that unique resistance pattern, get all these segregating offspring, and then see what the resistant seedlings have that the susceptible ones don't, to be able to map and position the placement of that resistance gene in the genome. And so Jason did that. He led the way in publishing. And then he found some SSR markers in the region that seemed to be linked to RDR3 relatively closely. And that's what's depicted here on this probably hard to see table in your meeting room. And so there's 3 different SSR markers by the 3 rightmost columns. And then the roses that have resistance to race 8 are depicted, and then those that were positive for the SSR markers are listed here that should have their resistance due to RDR3. So SSR markers are harder to run these days. The equipment's difficult to maintain. We wanted to convert to CASP or PACE markers. And then Jason had some old ion torrent sequence data with 3 roses resistant to RDR3 or having RDR3 with resistance and 3 susceptible. Deanna took that sequence data from Jason, and what Jason's mentoring was able to find SNPs within that region that the resistant ones had that the susceptible ones didn't. We took 48 of those promising SNPs, ran them as CAS markers across the original population. 12 of them still showed promise, and that's what I've been running here at River Falls. And I surveyed hundreds of different cultivars, some of which are susceptible to all the races, some that have the resistance pattern of RDR3, and we were able to go from those 12 promising SNPs, bracket the region down to just about 100,000 base pair with 3 SNPs. I'm at the point of needing to validate a few more roses, and then we'll have the data set ready to write up. One thing that I'd love just to highlight that's of interest is that one of those roses from the SSR markers that we studied and did the phenotyping on, folk singer, really opened a door for us to be able to bracket it down to those 3 SNPs as CAS markers. So folk singer, its pedigree is known. One of the parents then I was able to recognize has the RDR3 pattern, has the marker, go back multiple generations all the way to Goldilocks from 1945. And Goldilocks has the resistance pattern. I got it from Foundation Plant Services, did the phenotyping, did the genotyping. It was bred by Gene Burner, known as the father of the modern Floribunda roses, came out in 1945, one of the most popular roses of that time, the first yellow Floribunda that was widely grown and head above shoulders and performance and rich yellow color. So going back to Goldilocks and then following pedigrees back out, I was able to get a number of descendants of Goldilocks, phenotype them, find those that are candidates for RDR3, and then be able to then, through the pedigrees, find crossover events that were able to then bracket that region down to that finer region to give us those 3 very promising markers to publish on. And that's where I'll end here, except for one more little detail. Goldilocks, its pedigree is an unnamed seedling by doubloons. I got doubloons. It's susceptible to everything. So the original source of RDR3 and Goldilocks is a mystery. I got the biography of Gene Burner. There's no clue in there about it, so we may never know. So that's where the story ends for now. Thank you. And I'll stop sharing so Seth could take it over with his version.
Can I ask a question?
Go for it.
The cultivars that we're evaluating in the extension trials, I wasn't involved with the first SCRI grant, so I don't know much about the background of those cultivars and how they were selected. But do we know, as far as black spot is concerned, do we know what races are those potentially resistant to or not resistant to? Is it somewhere?
Hey, Dave. I don't know the results. That's Dave.
Ask David what he knows.
Did you hear that, David?
I have some data that's not shared yet. I gather every rose that I could that I either buy or propagate from cuttings or are given, and many do overlap. So I'm gathering data. So at least we'll have the phenotypic, data that we can infer to the RDR genes. That's a good question. We don't know what races are in the fields. Because from my understanding, we didn't isolate them and put them on the host differential. That would be a great project. And if I understand right, Oscar, your student Navdeep is pursuing that, which would be very valuable. And then over time here, we're beginning to understand some of the roses that were part of past studies may have some of the RDR genes that we're interested in.
Yeah, because I would be curious to know if the lack of resistance that we're seeing in the field corresponds to a specific race of the fungus. And so, yeah, maybe we can follow up on that.
Yeah. I have a question. David, Francisco here.
Hi, Francisco.
Hi, how are you?
Good.
I'm curious about how do you track in your progenies the three markers? Do you have PCRs for each of them, a multiplex PCR? What is the tool you use for tracking them?
Good question. So So we want a SNP-based marker. So we started working with the company IGC and their CASP technology, and they're pretty expensive. So we transitioned some employees from there, moved over to 3CR, and they have a cheaper, actually, it sounds like more robust system with better master mix. So CASP paste markers are basically the same thing. So that's what I'm working with a quantitative thermocycler PCR and just fluorescence.
Okay, thank you.
You're welcome.
Any other questions for him before I give some quick updates? Okay. So we've been talking about this a lot. You've seen this a lot. RDR4 is one that we are also very interested in because it gives us resistance to 13 or 12 out of the 13 races. This was found in bright eyes. And so last year, I submitted a HRI grant to do some additional research building off of what we've already learned. RDR4, again, came from, I believe, the rose breed from Jason Zern and them. They identified it, and I'm just trying to narrow down the region. As I said, Jason Zern and them have previously identified a 9-cent organ region on chromosome 5 that's associated with this. If that is a relatively large region, so I'm trying to narrow that bit so that we have a really robust marker that people can use into the future. So for this HRI, we're using multiple techniques to get at narrowing this region. Right now, I am working on a partnership with some collaborators in France that have some great resources that they can do some rudimentary genome assembly for us at a pretty reasonable cost, which is I was very excited to see because HRI grants, while they're wonderful, limited funding. I'm working with them. They are helping to develop a draft genome of bright eyes. We've started extracting DNA over there. And then the next step in Minnesota and Wisconsin, we're going to be doing an RNA-seq experiment looking at specifically the resistance response that we see in bright eyes. So I'm currently preparing, we're trying to get the plants free of disease in the greenhouse. Powdery mildew has been a little bit of an issue and we can't spray anything on them otherwise it'll interfere with our assays. And then finally, we're doing some genotyping by targeted sequencing of the known region so that we can really saturate the area with markers and have something really tightly linked, hopefully. So I'll go in a little bit more with the RNA-seq experiment just real quick, and then talk about PBTS. So the experiment setup is pretty simple, so to speak. So we have bright eyes that we will inoculate with both a race 12, which it is susceptible to, and then race that it is resistant to. We previously had identified race 10, but due to some fluctuations in how it's growing right now, we can choose a different race and then use a mock water control also. And we'll evaluate this at 7 time points after inoculation. So into each box that we have, we'll have a control. We'll have 3 replications within each race, and we'll do this over multiple time points. And then we'll collect tissue from these and hopefully be able to extract RNA and have that processed. So ultimately 49 samples of comprised of 3 leaf punches each, collected into RNA-free tubes. Yeah, and then additional backup samples. We're going to be doing some of this analysis at the University of Minnesota Genomics Center, maybe. If anybody is well versed in RNA, though, this is new to me, so would love to have some discussions today. And then hopefully this will result in anchoring and proposal of candidate genes combined with the genome work that is being done with some collaborators. So we'll have coding regions from the RNA-seq that can help assemble some contigs into longer scaffolds for genome development, and then can help you identify upregulated genes to propose some candidate genes responsible for this resistance. And then GBTS, as David said, 3CR has been a great collaborator for us so far. And they do really great work, and so we're hoping to use them again. They have started to pioneer some of this technology. So hopefully we can saturate this area with 20 to 40,000 probes. And then combining again with the genome, we'll be able to narrow in very much on this. And then we'll have our seedling population with our parents, along with ultimately resulting in SNP selection and conversion to PACE that anybody can will be free to use. So
this is the monthly test of the emergency notification system. No action required. This is the monthly test of the emergency notification system. No action is required at this time. This is only a test.
Okay. All right, we're all safe. So ultimately, we'll have Bright Eyes genome marker tightly linked that can be used by breeders, and then proposed gene of interest, and of course some manuscripts. This is all still in the works, so it's still very preliminary. And then for RDR5, we're on the opposite end. We have done a lot of phenotyping for this already. Ages ago. So this is all done by Diana, who you saw on that first slide. We have a population of Morden Blush and Ramblin' Red with 94 seedlings generated. They did detached sleep assays and were able to score resistant and susceptible seedlings that were then submitted for genotyping later on. And here you can see the resistance pattern for RDR5. Which is susceptible to 3, 10, 12, and 13. And so that population that I just mentioned was genotyped using the axiom array. A linkage map was developed off of that filtered. There were filtered and dosage scoring with fit poly. Integrated phased map was developed using polymap R. Ultimately, to identify, and I should have made this bigger because this is the more exciting part, RDR5 region on group 7 down here in the right corner, which means that this is the area that we're looking for that is tied to RDR5. So right now we've confirmed the region associated with RDR5. Identification and validation is in progress for the SNPs in the region, which we're working with 3CR to convert into PACE markers for wider adoption. And I know that they for those who are interested to learn more about this, because again, this was mainly done by Deanna and David and the team. The manuscript should be submitted soon. So hopefully within the next, 6 to 10 months, it'll be published, and you'll be able to read more about it. Which brings us to our last one, which is RDR6, which also confers some unique resistance. This project was interesting. So this was done as part of Emily Moore's master's thesis. Again, working with Diana and David and Stan. And they used an interesting approach. So they compared the loci identification via genetic map, so SNPs from the axiom array, and via bulk segregant analysis, which is essentially on the side here, you'll see we have our seedling population. You take the resistant, you take some of the susceptible, you genotype, you pool those samples, and then you genotype them, and are able to slowly, through process of refining your data, identify the region, hopefully, which they did. So both the axiom array and the BSA-seq techniques identified the same region. However, the BSA-seq was comparatively cheaper, and that's kind of what we were aiming at, is trying to identify technologies that can help us get to these regions faster and cheaper. So in addition to that, there was a comparison of developing mapping via the SNPs from the axiom array and the BSA-seq. Overall, you can see that the cost via the bulk segregant analysis was much cheaper. It was cultivar-specific SNPs, which could be good or bad, and then a larger confidence interval. So where are we at with RDR6 right now? Again, we've confirmed the region via these two distinct methods. We've identified 25 SNPs in that region, and validation is underway in a larger population of ancestors. And then we're currently working on converting these to PACE markers, again, for wider adoption in the future for breeders. And then there is a manuscript that's comparing these methodologies for submission, hopefully publication soon. And of course, one of the outputs was that Emily Moore graduated and this was accepted. So that's always great. Yeah, we love when students graduate, right? So with that, here's kind of a summary slide of where these 4 different markers, 4 different loci, pardon me, are at right now in our projects. Would love to talk more at lunch if people have more specific questions. And if I don't have answers, I can find out for you. So thank you.
And questions for Seth?
Yes.
Seth, bright eyes is almost impossible to find on the retail side.
Yep.
And for at least 3 years, it's been the top player in all this black spot research. So how are you getting the plant? Is David raising them in 5 different university greenhouses?
No, David, though, has connections to a lot of Rose people, and we have been able to get them through one of his sources, and I'm blanking on the name. If David's still on, feel free to chime in.
We were able to get cuttings from Foundation Plant Services and then also STAR. So even though it's patented yet, we were able to get permission. So that's been a big limitation. I agree. It would be great for everybody to have access. It would be great for it to be reintroduced commercially.
Have you talked to Brad Yoder about that?
No.
Keeping it on the market?
Nope, I haven't.
Okay, we're going to talk to him.
Okay.
All right. If not, I don't want to keep you long.
One more question?
In the field, we have seen a lot of black spots, so we don't know what races we have in the field. So is there any possibility we can find out?
That's a great question. And I think that goes back to maybe a little bit of the earlier conversation that was happening at the end of David's. So yeah, maybe if we can figure out what's in the field. I'm looking up here because
Yeah.
well, we might hear more about that later. I don't know. A little bit, okay.
And then, we see a lot of variation in symptoms in maybe the cultivar interaction between the fungus. In some cultivar, really things marching and then circular kind, but in some really diffuse, but really tiny spot, but like that kind of symptoms, maybe that variability maybe due to the interaction of different fungus or different races.
Could be, and it could also be
Yeah.
quantitative resistance,
Right.
seen in some of these things
No.
versus these.
Are you saying that, hey, can we send samples to deep to sort out the races?
So I'm gonna make a comment right now. Nobody is doing race work on roses like they used in the past.
Mhm.
So all of the stuff that we're seeing now is things that started a long time ago that's now getting finished. So that is why we're working with Kevin on, among other people, to try to develop a method to try to scan for black spot races under field conditions through sequencing approaches. Now that is a tool we're developing, but it's not available, but it would be a way to get at how many races of black spot do I have in my field. You do a random statistical collection, you sequence, and then hopefully you'll know. But that is the only thing we can do now because we just don't have a plant pathologist that is willing to do race work for the community, right? So that is one of the things that we're trying to do. And we're trying to do the same thing for Cercospora lispot, as you know. Okay. But it's still trying to be developed.
I'm just gonna say hi here real quick because David's gonna take this the intro here.